Hi, my name is Kitana Lepardo, and my project for this year's symposium is called can Using CRISPR to cure sickle cell anemia can alter its chemical structure. So a brief synopsis about sickle cell anemia is that it is a rare blood disorder, one of many anemic diseases. And why it is called sickle cell anemia is that the red blood cells or erythrocytes generally have a sickle shape, and it is just basically irregular or misshapen red blood cells. That is due to the lack of oxygen from the hemoglobin. The hemoglobin would not be able, basically is basically unable to transport oxygen throughout the body, and that hemoglobin is consisted of a heme iron group, and within that heme iron group is where the oxygen is meant to bind to a heme iron group, which is why the hemoglobin is able to transport it throughout the body. However, with sickle cell anemia, they're unable to do that, which is what is resulting into its irregular shape. Now, luckily, with the invention of CRISPR and the two branches of CRISPR, Casgevy and Lyfgenia, there are two different types of gene therapy tools that are able to basically go into the gene, modify what is creating the mutation of that hemoglobin, and alter it, change it however they like into another type of hemoglobin, and basically transplant it back into the bone marrow, allowing it to undergo replication and create more of that protein. And my overall research is to basically see if that modification of the hemoglobin would alter its chemical structure and basically impact the iron absorption levels within the body. Now, what you see here at the bottom is the hemoglobin chemical structure, which is the heme iron group. So you can see that porphyrin rings right here. In the middle is the ferric state, which is Fe2, and that is generally where the oxygen is meant to bind. And once the iron is oxidized, it becomes the ferrous state, Fe3 plus, and yeah, we'll be able to transport oxygen throughout. Now that porphyrin rings is basically meant to go into here, which are the four subunits, beta 2, beta 1, alpha 2, and that is within that hemoglobin still. And as you can see, since there's alpha and beta subunits, that is basically what consists of an adult hemoglobin, which is something we all have. So, yeah. This is the hemoglobin itself. The heme iron is basically going through heme synthesis. Yeah. So, the main purpose of this experiment is generally to, of course, see the chemical structure. Now, unfortunately, I don't have CRISPR genetic tool in my backyard, so I do have a microscope instead. So my whole purpose of this experiment is to basically see, is to allow the people to visualize the irregular shape of someone who has anemia or sickle cell anemia of their blood cells. So I was able to use a GIMSA and right stain and see here blood platelets that is caused by the wound of the red blood cell of the person that is creating that generally just prick their fingers and get the blood. And see is probably granules, basophils, neutrophils, leukocytes, but most importantly, you'll see irregular shapes. These are not all regular disc shapes. They're not similar in shape, uh, especially to this one right here that has almost like a sickle-like shape, almost to sickle cell anemia. Now this would have been sickle cell anemia if they had actually half-shaped or C-shaped red blood cells, but you generally get the gist of it. Just to give you an overall idea of what it would look like with someone who has a lack of oxygen being transported. Now, there are two different types of hemoglobins, adult and fetal. Now, I did talk about adults. There is, which is basically the common hemoglobin. There is fetal hemoglobin, which generally has a higher affinity to oxygen and therefore has a higher iron absorption level as a heme group. Although there is heme groups, there's also um, a non-heme group, and those are generally being consumed. It's just us being like consuming medications such as ferrous sulfate, probably uh, plant-based foods, stuff like that. Basically a type of iron that is not being generally created within the body naturally. So with Casgevy, also known as exodenvogene autostem, it basically, as I said, modifies the blood stem cells, transplants them back into the bone marrow, but typically it is encoding the gene to become a fetal hemoglobin because that means that it's generally for those who have sickle cell anemia, so that since fetal hemoglobin has a higher affinity to oxygen, that is basically to counteract that. However, with lipogenia is with those that do not have sickle cell anemia. It's for those who do not have sickle cell anemia, but another type of blood disorder, so just a regular anemia. And it's basically meant to produce HbA T87Q globin, and basically a synthesized version of the adult hemoglobin, but have a special effect, and they basically have the ability to reduce the chances of having a sickle shape of an adult hemoglobin. Now, as you can see here, as you can basically see,